Hello everyone, welcome to GPAT again. So this is the recorded lecture class 2 and this will cover the quick revision of analytical pharmacognosy last portion. So in the previous video we have seen that we were discussing about the evaluation of drug, that is the main portion for the analytical pharmacognosy, and the drug evaluation consists of the morphological, microscopic evaluation, the physical evaluation, chemical evaluation, and the biological evaluation also. So in the organoleptic evaluation, we were seeing that drugs are determined by their size, color, shape, etc. And we are continuing the microscopic evaluation. In that, we have already covered the stomata and trichomes before. And now we are going to cover the calcium oxalate crystal. So this is one of the most important part for your exam, calcium oxalate crystal. You can see questions from there, and in the previous years also multiple number of questions were coming from the calcium oxalate crystal. Just like the stomata and trichome, it becomes important for you. So the classification is, there are multiple parts of a multiple number of classification for the calcium oxalate crystal. So firstly there comes the prismatic or cubical, and all are written here with the tricks. Okay, so if you remember the tricks, then you can remember the examples, and mainly from the when you are continuing your main drugs, okay, like the cascara, coriander, so in that also you can remember from this tricks, and mostly from the examples the question comes. So the trick is, Q Queen Kuchi and Sena loves handsome and witty Rahul. So cascara, coriander, CQ actually it is. So CQ is the CQ. CQ means what? CQ is cascara, coriander and cardamom. 3C. 3C is the CQ. CQ Queen Kurchi. Queen is Quelia and Kurchi is Kurchi. Sena loves is licorice. Handsome and witty. Hyoscyamus rolfia wild chili. Rahul is rolfia actually. Hyoscyamus and witty. Witty is wild chili. Rahul is rolfia. So Rossetti and cluster type crystal, every classification for the calcium oxalate crystal is equally important. Okay, you can't leave anything because all are important. And when you are pursuing your drugs, you can see also that for the examples, the calcium oxalate crystal type comes in the microscopic evaluation part in your particular drugs. So you must remember the classification with examples. So Rossetti or cluster type is the rhubarb and clove, surely fassa in Facebook. Rhubarb, clove, surely is Sena, fassa, Punarnava, Hyoscyamus, Arjuna, Stramonium cascara, anise. Facebook is fennel, eucalyptus and cascara. You can see the bold words for your initials of your drug name. So rhubarb, clove, senna is surely. Fasa, PHA is a. Punarnava, Hyoscyamus, Arjuna, Stramonium and anise. Facebook, FCE represents fennel, eucalyptus and cascara. The Raphides is for the Ras in Germany country. Ras. Ras is for the Rolfia, Andrographis, Aloe and Squill. Germany is Gentian and country is cinnamon and ipecac. Microspheroidal or sandy crystal, this is very important. Datura ne bela ho सिंकोना में highly danta. Datura, bela is belladonna, सिंकोना highly is henbane and danta is duboisia. And there is also tobacco. I can match the tobacco one. So you remember the tobacco also in the microscopical or the sandy crystal. For the tetragonal, it is to. As you can say, to kya hua? So that to is for the tetragonal, and O is for onion and H is for Alzheimer's. Monoclinic crystal is for the Vietnam Queen Oak, Veratrum oak gall and Quelia. Next is rhombic or diamond crystal. So this is the Tulsi Rani Kurchi, wears diamond. So Kurchi Mimosa pudica. Mimosa pudica is the Tulsi and Rani is rhombic or the diamond, and Tulsi Rani Kurchi wears diamond. Needle shaped crystal is the Phytolacca americana. This is the example. Next is quantitative microscopy. These were the qualitative one. The stomata or the calcium oxalate crystal or the trichomes, they were the qualitative microscopy. So with the quantitative microscopy, we see the lycopodium spore method where there is the percentage purity of drug is calculated by the particular formula, and that number of lycopodium spore in the standard is the 94,000. Next is the chemical evaluation. So our microscopic and morphological evaluation is completed with the note, and the chemical evaluation contains the bitterness value at the standard is quinine hydrochloride, and with this standard 1 gram of quinine hydrochloride the bitterness value of drug is determined. For the hemolytic index, the reference material is saponin. Okay, the reference material is saponin. For the determination of tannin, tannin reacts to protein because of its astringent action. It will form the insoluble tannate which are resistant to action of proteolytic enzyme. So basically the astringent action of astringent action of tannin is essential for precipitating the proteins. Okay, tannin precipitates the protein. So this is the main criteria. For the pesticide residue should not be more than 1%, and for the heavy metals there are some limits. For lead 10 mg per kg, cadmium 0.3 mg per kg, these are the limits for your evaluations. Determination of foaming index. Okay, foaming index is 1000 by A, and foaming index is basically the amount of foam that is created by the plant material when there is 100 ml of water or 100 gram of water it is dissolved into. So A is the volume in ml of decoction used to preparing the dilution in tube where the foam of 1 semi height has been observed. When the cellulose sailed and treated with ZnCl2 and I2, I have always told that when there is iodine, there is blue to violet color. So it lignified cell wall with fluoroglucinol. Fluoroglucinol is always pink or red color, red or pink color. Fluoroglucinol, you have seen already in the previous class where we were discussing the questions. There you have seen that there was one match the following with the reagent name and its color. So both of the ZnCl2 I2 and fluoroglucinol were there. So I told that when there is iodine, there is blue to violet color. When there is fluoroglucinol, there is red to pink color. Starch with iodine, always blue color. Halfen's test is for cottonseed oil. Van Ark's reagent test for argon, and Vitally-Morin test is for the tropinol colors. Next is the physical evaluation. So the physical evaluation is not that much important, but I think you have all covered it in your practicals. The moisture content determination. Moisture content is determined in every 1 hour actually, and it is placed in the hot air oven. For one plant material is weighed, and by putting it in a petri plate, it is put into the hot air oven, and in every 30 minutes or 1 hour, the weight of the plant material is taken, and after 3 to 4 hours the weight taken, when the weight becomes same after 1 hour also, like after 3 hours you are getting 0.5 mg, and after 4 hours also you are getting 0.5 mg. So when that is constant after a certain time, then that will be the final weight. So initial minus final weight by initial weight into 100% with the moisture content in percentage. Viscosity is also determined by viscometer. Melting point determination, solubility determination, and ash content, physiological ash content determination, the hydrolyzable ash content determination. You can see in the PDF also that I have already given when our regular batch was continuing. So you can see from there. And the fluorescence analysis, this is one important point. And for the fluorescence analysis, you have different drugs, and basically from this drugs question comes. So that is why the drugs are given here with the fluorescence color. So cinchona is giving purple blue, gentian root is whitish blue, ipmoya is deep purple violet, quassia is whitish blue, rhubarb is violet, pale catechu or gambier plant, it is giving green fluorescence. In the gambier also, when we are continuing the resin portion, so there also you have seen that pale catechu and black catechu, what their differences are, what is gambier and what they're containing. So quercetin was a huge portion, and the fluorescence color also you have seen that. So Glambier fluorescent test was also there for the identification of the pale category. So that is the green fluorescence it is giving. And Cassia cinnamon is giving green blue fluorescence. Terminalia arjuna is pink. Tomentosa is giving blue. Rapnotic rhubarb, Indian rhubarb, Chinese rhubarb. These three are very important. This last of this, sorry, last of this are very important, and that is why I have given in multiple mock test question from there. And rapnotic rhubarb is giving blue fluorescence, whereas Indian rhubarb is giving violet fluorescence. The Chinese rhubarb is also giving blue fluorescence. Argometrin is very important, okay, very important. It is also giving blue fluorescence, and imetin is giving orange fluorescence. So these are the fluorescence portion, and last here comes the biological evaluation. And the biological evaluation is checked by the hepatoprotective. Basically the pharmacological action are mostly determined by the drugs, and this is related to the biological evaluation. This is not that much important, but if you want to know, you can pursue it from his textbook. So this is up to this recording, and on the next day we will cover some another part of the pharmacognosy in our recorded lecture with the quick revision. Thank you everyone.